Hey guys, it's Professor Tiffany Martin with Medical Microbiology, and we are working our way through chapter 16, and we need to define what T cells are. So we're at the third line of defense, the adaptive immunity specific. We said it's going to involve T cells and B cells. So let's talk about T cells for a few minutes. Okay, so T cells. T cells are going to work with the cell-mediated immune response. They are designed to attack pathogens that are already inside a cell. So they are going to work against intracellular pathogens. They are going to identify those endogenous antigens that when the pathogen gets into a cell, the cell is going to take some of those proteins from the pathogen and put it on the outside of its cell as a red flag, going, hey, look, I'm invaded, come help me. These are what the T cells are searching out. They're looking for cells that have a pathogen within it. So that's what our T cells do. They're T lymphocytes. And they're going to involve with that. And this is called a cell-mediated immune response. All right, so they not only look for cells that have a pathogen within, but they'll also look for cells that are not behaving correctly, that are producing abnormal proteins. These could be cancer cells. These could be our own body cells that are not working well and they need to be targeted and gotten rid of. That's also part of the T cell's job. So they will defend us against that as well. Where do we find T cells? T cells are made, right? They're made in the bone marrow. We know you've told me that they mature in the thymus. And then once they go through the thymus, maturation in the thymus, then they go out into the blood and into our lymph system. So that's where we would find T cells. All right, so how do they recognize these antigens? How do they bind to these epitopes? Well, these T cells are going to have something on their surface, on their cytoplasmic membrane called T cell receptors. So what's a T cell receptor? T cell receptor is going to be, let's go to the picture and I'll come back. A T cell receptor is going to be a protein on the surface of a T cell. So there's my T cell membrane. This is my protein that we are identifying it as a T cell receptor. It's got a portion of it that anchors it into the membrane, so it's an integral protein there. It's got a large portion on the outside of that T cell receptor. It's going to have a place on that protein that's going to bind to the antigen, specifically the epitope. Remember, we defined antigen. We watched that video. Antigen's a protein that's on the pathogen. The epitope is the specific amino acids, the little specific area that binds, and it's going to fit right here against this T cell receptor. Now, there's going to be some regions that are the same for all T cell receptors, and this one T cell is going to have a lot of these T cell receptors, and they will all be identical on this one cell. Now, we will have a different T cell that will have T cell receptors, and this part will all be the same, but each T cell is going to have a unique antigen binding site that's going to allow it to undergo the specificity part of this third line of the defense. It is going to have a target that it's looking for. It's looking for the antigen, the specific antigen that fits right here in this T cell receptor. So that's how you get the specificity. That's how you get these unique T cells that are designed to fit against one specific pathogen. All right, so back to our word slide. Our T cell receptors, the specificity of the T cell receptor comes from that TCR, the T cell receptor. Each one has that specific antigen binding site. This is going to be variable. Now, let's talk about this variability first. So this variability, we've got, here I'll go back to the picture, we've got this protein that's mostly the same in all T cells, but this part has to be different, and it has to be unique for all T cells. So let me talk about how we generate that. We know proteins are encoded in our DNA. We have an instruction manual, an instruction on how to build that specific protein. So in order to get T cell receptors to be specific, we have to have a different place, a different series of amino acids right there at that antigen binding site. So if in our DNA, if we tried to make a new instruction or an instruction section for every single T cell receptor, we couldn't make that many. We just don't have enough room in our DNA to make that many proteins because we've got to have our T cells ready to go against any potential pathogen that comes in. And there's a ton of them out there. All these different bacteria, all these different fungus, all this different, all of this stuff out there. We need to make sure that we have a specific antigen binding site for that specific pathogen. And that's just overwhelming. There's billions and billions of different pathogens and different types out there, and we've got to be ready for any of them. So how do we get that variability? So we are going to do that by doing some mixing and matching in DNA. So let me draw that out for you. We're going to have our DNA here. There's my line of DNA. And we're going to have a gene on our DNA. We've seen that before. So I've got a gene, promoter, terminator, remember all of that. So you can make mRNA so that you can make the protein. So I've got a gene in my DNA here. And this gene is going to be for a T cell receptor. And now again, I don't have room to do a whole bunch of T cell receptors. So I've got one area that's going to be a T cell receptor. And in the gene, I'm going to have a lot of different instructions. So a lot of different areas in this gene. When it gets transcribed into an mRNA, instead of starting at the beginning of this gene and going all the way to the end, it's going to pick up a little section here, a little section here, and a little section here, and make an mRNA just from those regions. This one, and then this one, and then this one, and you put those pieces together, and this is going to be your mRNA. Okay, so that's what actually gets transcribed and then translated to make our T cell receptor. So if I need to make a different one next time when the RNA comes through, it's going to take a piece here and a piece here and a piece here and put that together to make a completely different mRNA. And it can do that by using these variable regions and some other stuff like that. So when we do our transcription, this mRNA is completely different from this mRNA, and this is how we get variability. So they're just different regions in this gene, and you can mix and match these regions. I can do another one, and maybe I pick up this region, but then I pick up a blue region here, and then maybe there's another region down here, and I pick that one up. And so now I have a third mRNA that's completely different from the first two. All right, so this mixing and matching creates the variability. If you want to wrap your head around it, think about your closet and think about having 20 pairs of 20 shirts and 12 pairs of pants and 9 pairs of shoes, and you need to make a different outfit. And so you take one of your shirts and one of your pants and one of your shoes and you make an outfit. The next day you can take a different shirt, you can wear that same pair of pants and a different pair of shoes, and people will think you have a different outfit on, completely different outfit. So if we do that enough, we can get variability and we can get to those billions of different possibilities of pathogens coming at us. So when you think about this, so this one I drew, this is the instructions you got from your mom, but you have another set from your dad, and that's going to have purple, orange, and pink. It's going to have completely different ones here, and you have those possibilities to make those outfits or those mRNA. That's going to get transcribed into your T cell receptor. So this creates a lot of variability. Now there's going to be some similarities. Let me go back to the picture. There's going to be some similarities in that. This transmembrane portion, these constant regions, these get worn no matter what. So this is your part in your closet that you wear no matter what. But then this variability comes from picking from a couple of different regions from that. So this is how we can create these unique T cell receptors so that we can be ready for any potential pathogen. Okay, so ask me some questions about that in Zoom. Specific site is due to those variable regions, that picking and choosing from DNA that gets transcribed into different mRNAs that create that unique, unique as a snowflake, you never have to repeat an outfit, T cell receptor from that. Okay, the next thing I need to talk to you about is that these T cell receptors do not bind just to the epitopes. if I have an epitope, so you remember that's the piece on the protein. Here, I'll go back a couple of slides. Remember, the epitope is the piece of the antigen that is going to get bound to a T cell receptor. The T cell receptor can't just bind to this as is. It actually needs some help, and that's going to come up with the MHC proteins and the major histocompatibility proteins. So we will do that on the next video and figure out how these T cell receptors actually bind to those antigens. Okay?